Genomics has the potential of really revolutionizing personalized healthcare. We will eventually sequence every individual in the United States. In using this information, we will be able to predict what diseases they're likely to get. One of the deficits of the genome-wide association studies that have been conducted to date is that they've been largely conducted using individuals of European ancestry.
When we think about ancestry, we tend to think about different categories. So we might think of someone as being white or black or Asian. But in fact, many people, including a significant portion of the US population, don't fall neatly into those categories. They're actually admixed, meaning that they draw portions of their ancestry from multiple different groups. For example, if we think about African Americans, many African Americans tend to have ancestry both from a European component as well as an African component. Many of us are admixed, at least to some degree.
CAST is a Center of Excellence in Genome Science funded by the NIH, and its focus is on leaving no one behind. Currently, admixed individuals are not represented well in genome analysis and studies.
A goal of CAST is to look at an individual and take into account their unique mixture of genetics and ancestry and socioeconomic factors, as well as environmental factors. And how can we take all that into account to build models that are really accurate for each individual?
The difficulty in using the causal SNPs or predicted causal SNPs that have been identified in genome-wide association studies to date is that these studies have been largely conducted in Caucasian populations. Because the structure of the genome, the linkage disequilibrium structure of the genome is different between different ethnicities, it's difficult to translate the information that we found in these GWAS studies into other populations, particularly admixed populations. It's really impeding our ability to translate the fruits of these genomic studies into personalized healthcare for all Americans.
When we start to think about ancestry and admixture in this way of these different mixtures of these different components, then ancestry becomes much more of a quantitative type of a variable compared to a categorical variable. And so now those distinct categories kind of no longer make sense, and we instead really need to think about ancestry as being along a continuum. Tandem repeats consist of repeated units of one or more base pairs in a row. There's something like 2 million different tandem repeats across the genome. So SNPs we often think of as being biallelic, meaning there's just 2 common alleles in the population. It's kind of like a switch. Rather than treating it as a switch, we treat the repeat as a quantitative variable, and we can look for associations between repeat copy number and traits, and we've now identified hundreds of these associations between repeat copy number and things like expression of nearby genes. Those associations that we're finding often turn out to have really ancestry-specific patterns.
We have methods that take this into account to analyze not single SNPs, but HLA types, which are in general haplotypes that cover one HLA gene. They include all SNP genotypes that are inherited together. We have an improved accuracy for genotyping and therefore to perform genome-wide association studies, and this will, in the long run, improve the development of polygenic risk scores in diverse populations.
Social determinants of health, abbreviated as SDOH, refer to those non-medical factors that influence health outcomes, such as the conditions in which people are born, grow, live, work, and age. SDOH is estimated to account for about 30 to 55% of health outcomes. We need to formally represent those SDOH factors and standardize their values in order to include social determinant health into the quantitative analysis of phenotype-genotype association studies. That's why we're developing ontologies to formally represent SDOH information.
We're working closely with the All of Us research program by the National Institutes of Health and the Million Veterans program by the Veterans Affairs. These programs are some of the largest genomic data collections in the United States, and they include individuals with diverse ancestry and sociodemographic backgrounds. The problem is that due to the sensitive nature of genomic data, these data are often put in silos and not shared for research due to policies and regulations that limit the scope of data sharing. And to address this, we're developing computational solutions that can help us share data across silos while protecting privacy of the underlying individuals. And we're working to further develop our tools and make them more broadly available to other researchers.
Currently, people self-reported race includes concepts such as the genetics involved in it, but many times also the social determinants of health that are more frequent in certain races. So in order to disentangle the concept and not rely so much or at all in self-reported race, we can study genetic determinants of health as well as social determinants of health.
Another challenge that we really face is looking at places where ancestry currently already is being used in some of these models, but it's being used incorrectly in a way that might actually be harmful and adding to existing disparities between groups.
CAST is an inclusive project that aims at having genome science apply to everyone.
The goal of CAST is to be able to set aside categorizations of individuals into different ancestries and to be able to jointly analyze them and also be able to use these findings to predict the relative risk of everyone in the United States, regardless of their ancestral backgrounds.